Welcome to our next lesson. We should have completed our experiment last week with the last lesson, sorry, with the vinegar and antacid. So we're going to have a look at the analysis of that experiment today. So today's tasks, you want to analyze your data, and what that means is do a quick write-up. You want to complete your online quiz for the next week, and we'll have a quick talk about catalysts at the end. So here's our data table from last time. You should have some information written in here. If you haven't got some information from doing the experiment, I suggest you shoot a message to one of your friends and see if they can provide you with the data. So we looking at the surface area of the antacid, a whole antacid versus a crushed one. We looked at the temperature. So we put the vinegar in the fridge versus in the room temperature. And we looked at the concentration of it, as in just straight vinegar was the first one, and then diluted half and half with water in the second one. And what we should see is that the reaction works better in certain circumstances. The crushed tablet is going to have a higher surface area, so the time taken should be reduced. The height reached should be a lot bigger than the other one. At room temperature, there's more energy in there because it's warmer, so hence you should have a higher height here than in the fridge version. And in the concentration, this first one here is going to have more vinegar to react, so it should reach a higher height than the second one. That's essentially the summary of what should have happened. We had these questions before. Have a look at those. Make sure you answer them as a bit of a prac write-up. And if you look at the PowerPoint and the video from when we first talked about this experiment, it gave you the sort of outline of how you can write it up. You normally stick your title, you have your aim, there was a little hypothesis section, your materials and so on. It's usually a good idea to draw a picture on something like that. And then you have an analysis section where you address those points, and a conclusion where you address those ones there. So your task is to get those things written up for this lesson. Other things you need to do. You've got two little forms to complete there, two little quizzes. The first one here is a quiz, and the next one relates to the experiment that we've done. So you have to answer a quick question about that. But if you've done your write-up on the previous slide like I told you to, then this second task should be fairly straightforward. The other task to complete today is a quick investigation of catalysts. Recall a catalyst is something that's used to make a reaction go faster. However, it's not used up in the reaction itself. Something like adding more oxygen will make wood burn quicker, but the oxygen's used up, so it's not really a catalyst. Now, in a proper lab, you might use something like manganese dioxide and hydrogen peroxide. So peroxide you can sometimes find around the house. Manganese dioxide is probably a bit harder to find. And the idea with this experiment is to add a bit of hydrogen peroxide in the bottom of your test tube, add some manganese dioxide to the top there, and look at the reaction of it. Look at the reaction in different, you put hot water and so on there under different circumstances. You can pause the video and read through the information on the screen there. Likewise with this one, you can do the same thing there. And there's a little discussion section. You should do these discussion questions as well. Now, if you don't have these materials and you're not in the lab, what can you do? You'll have to try alternatives. So here's some alternatives. You can do the experiment in the first point here with hydrogen peroxide. If you've got some around the house, you might have some in the workshop. There might be some in the bathroom because sometimes it's used to bleach things like hair and so on. Hydrogen peroxide, when you sit it on the bench, we've discussed before when we talked about the Big Bang explosion and the comparison. When it's sitting there on the bench, if you have a beaker or a glass of peroxide, eventually you see little bubbles start to appear on the outside, a bit like a soft drink, and that's the oxygen coming out because the hydrogen peroxide, which is H2O2, will break down into oxygen and water. O2 is oxygen plus H2O water. But you can add a catalyst to make it go quicker. Liver has an enzyme in there, a catalase enzyme, that breaks it down because peroxide is harmful to our bodies. Hence, any sorts of liver will have the catalase enzyme in it to break down the peroxide, because oxygen is useful to cells, water is very useful, so it's not a dangerous or a harmful chemical in our body like that. You can do the same reaction with potassium permanganate, which is Condes crystals, those dark purpley coloured crystals. Or if you have a sample of yeast in the house, just add a small sample of yeast, like not even half a teaspoon, smaller than that, add it into a small glass of peroxide, and you'll start to see them. The yeast one particularly, you'll see it bubble quite a fair bit, whereas the chopped liver is more like when you leave it with like soft drink sitting on the bench. If you leave soft drink on the bench, you'll see the bubbles on the outside. That's kind of like the chopped liver version, whereas the yeast is quite a fast, quite a rapid reaction, and you'll see it bubbling up. If you Google peroxide and yeast on YouTube, you'll find some videos of it. Another example of a catalyst is an enzyme in your saliva. You have the enzyme amylase in your saliva, and what that does is it breaks down starch into simple sugars. Starch is just basically a line of simple sugars, like a chain like this of simple sugars. It's like sugar molecules, just like that, quite long. And what the amylase does is it breaks the bonds between them in the starchy molecules, like this, breaks those bonds, and you release the individual sugar molecules like that, which your tongue will taste sweet like sugar. So if you get like some bread, get some nice white bread or something, and then chew a bit of it, and then just chew it for quite a while in your mouth. And when I say a while, I mean, 30 seconds type thing. And it'll start to taste a little bit sweet, and that's the amylase breaking down the starch into sugar molecules, individual sugar molecules, and you'll taste that. Another way to think about this, and it's not really a catalyst in terms of an actual chemical or enzyme or substance that you add. In this scenario, make up some hot chocolate, use cold milk, and use hot milk. And obviously when you mix up hot chocolate, you want the hot chocolate to dissolve in the milk. Hot milk, it will dissolve quicker. There's a lot more energy in there, which will help break down the particles and dissolve in amongst the milk. Cold milk has less energy, and much like our experiment that we wrote up earlier in this video, cold environments have less energy and the reaction goes slower. Have a go at those so you can do the experiment at home. And that brings us to the end of today. Have a think about what we did today. And what are we doing next time? We're going to talk about the reactivity of metals. Okay, thanks for coming.